A substance known under the trade name of warfarin was destroying rats, chiefly through its action on a blood anticoagulant and interference with vitamin K. Warfarin-resistant rats, mutant strains, appeared first in a locality in England and have since been spreading and replacing the ordinary warfarin-sensitive rats. Our struggle against diseases and insect pests is unending as long as the mutation process continually produces variants that may have survival value in an altered environment. 4. Antibiotic resistance in bacteria. Penicillin, sulfonamides, sulfa drugs, streptomycin and other modern antibiotic agents made front-page headlines in the newspapers when first introduced. These wonder drugs were exceptionally effective against certain disease-producing bacteria and contributed immeasurably to the saving of human lives in World War II. However, the effectiveness of these drugs has been reduced by the emergence of resistant strains of bacteria. Medical authorities consider the rise of resistant bacteria as the most serious development in the field of infectious diseases over the past 2 decades. Bacteria now pass on their resistance to antibiotics faster than people spread the infectious bacteria. Apparently, mutations have occurred in bacterial populations that enable the mutant bacterial cells to survive in the presence of the drug. In them, thus, mutations furnish the source of evolutionary changes and the fate of the mutant gene is governed. Staphylococcus aureus is among the many bacterial species that have evolved resistance to antibiotics. The evolution of such supergerms threatens to reverse R. By selection. In a normal environment, success in the battle against infectious disease. Mutations that confer resistance to a drug are rare or undetected. In an environment changed by the addition of a drug, the drug-resistant mutants are favored and replace the previously normal bacteria. It might be thought that the mutations conferring resistance are actually caused or induced by the drug. But this is not true. Drug-resistant mutations arise in bacterial cells irrespective of the presence or absence of the drug. An experiment devised by the Stanford University geneticist Joshua Lederberg and his wife Esther Lederberg in 1952 provided evidence that the drug penicillin or streptomycin permitted pre-existing mutations to express themselves. As shown in figure 6.3, colonies of bacteria were grown on a streptomycin-free agar medium in a petri plate. This is called master plate. When the agar surface of this master plate was pressed gently on a piece of sterile velvet, some bacterial cells from each bacterial colony clung to the fine fibers of the velvet. The imprinted velvet could now be used to transfer the bacterial colonies onto a second agar plate. In fact, more than one replica of the original bacterial growth can be made by pressing several agar plates on the same area of velvet. This ingenious technique has been aptly called replica plating. In preparing the replicas, Lederberg used agar plates containing streptomycin. Of course, on these agar plates, only bacterial colonies resistant to streptomycin grew. In the case depicted in figure 6.3, one colony was resistant to the drug. Significantly, this one resistant colony was found in the same exact position in all replica plates. If mutations arose in response to exposure to a drug, it is hardly to be expected that mutant bacterial colonies would arise in precisely the same site on each occasion. In other words, a haphazard or random distribution of resistant bacterial colonies, without restraint or attention to location in the agar plate, would be expected if the mutations did not already exist in the original bacterial colonies. In second set of experiment, Lederberg tested samples of original bacterial colonies in a test tube for sensitivity or resistance to streptomycin, figure 6.3. It is remarkable that the bacterial colonies on the original plate had not been previously in contact with the drug. When these original colonies. Spontaneous origin of mutation. Bacterial colonies imprinted on velvet remaining. Colonies on plate, bacterial growth in absence of streptomycin. Plate 1. Sterile plate pressed on, imprinted only one colony with streptomycin velvet. Colonies adhere, grows. Isolation and test of colonies. Original plate 1. All tubes contain streptomycin. Scent U. RL growth, no growth. Streptomycin resistant, streptomycin sensitive. Figure 6.3. Experiment of Joshua Lederberg revealing that drug-resistant mutations in bacterial cells had not been induced by the drug but had already been present prior to exposure of the bacteria to the drug, after Volpe, 1985. Were isolated and tested for resistance to streptomycin, only one colony proved to be resistant. This one colony occupied a position on the original plate identical with the site of the resistant colony on the replica plates. This experiment demonstrated conclusively that the mutation had not been induced by streptomycin but had already been present before exposure to drug. 5. Infectious diseases in humans. J.B.S. Haldane, a British geneticist, has suggested that infectious diseases have been one of the most potent agents of natural selection in human populations. It is difficult to prove clearly that genetic changes have occurred in the human resistance to pathogens during widespread epidemics in the past. Nevertheless, it is a reasonable supposition that major epidemics eliminated the selective survival and multiplication of individuals provided with greater genotype resistance. During the 18th and 19th centuries, tuberculosis spread with the growth of cities and created havoc throughout the industrialized world. It became known as the Great White Plague. Annual mortality rates during the industrial era were as high as 500 per 100,000. Tuberculosis or TB is a highly infectious disease. Minute droplets of sputum, discharged by the cough or sneeze of an J.B.S. Haldane, 1892-1964. Affected person, contain literally thousands of tubercle bacteria. Bacillus mycobacterium tuberculosis, which can survive in the air for several hours. Even thoroughly dried sputum, when inhaled, can be infective. TB may invade any organ but most commonly affects the respiratory system, where it has been called consumption or phthisis. In 1750, tuberculosis flared in crowded London and then swept uncontrollably through the large cities of the continental Europe. Immigrants from Europe brought the devastating disease to the United States in the early 1800s. The death toll was highest in cities with large immigrant populations: New York, Philadelphia, and Boston. As tuberculosis advanced across the nation, it became the chief cause of death in the United States in the 19th century. Tuberculosis then declined steadily in the 20th century in all industrialized countries. From a high value of 500 deaths per 100,000 in the 19th century, the mortality rate dropped in Europe and the United States to approximately 190 per 100,000 in 1900. By 1945, the rate fell to about 40 per 100,000. The decline in mortality cannot be attributed, in any large measure, to general advances in living conditions, or to therapy. The decline was in full force before effective medical steps were taken, and indeed, even before the discovery of the tubercle bacteria. The causative agent was discovered only in 1882 by Robert Koch in Germany. The available information suggests strongly that the reduction in mortality has primarily been the result of an evolutionary reduction of virulence of the pathogen coupled with a heightened, genetically conditioned, resistance level of the host. In other words, the severity of infection has most likely been diminished by genetic selection and has been essentially independent of medical or public health mediation. 6. Sickle cell anemia. The operation of natural selection can sometimes be rather complicated. A variant form of hemoglobin, HbS, in human beings is responsible for the genetic disorder or molecular disease Volpe, 1985 called sickle cell anemia, discovered by American physician James B. Herrick in 1949. Individuals homozygous. Robert Koch, 1843-1910. Gauss for the abnormal hemoglobin dye at an early age. Even in the heterozygous condition, the red blood cells containing abnormal hemoglobin become sickle-shaped, and are unable to bind oxygen efficiently. Why has natural selection not eliminated the abnormal allele? A clue to this puzzle is given by the geographical distribution of the abnormal hemoglobin. This variant is often found in areas where malaria is common. Detailed studies have shown that the malarial parasite spends a part of its life cycle in the human red blood cells. When a RBC in heterozygous individual becomes sickle-shaped, it effectively kills the parasite. As a result, individuals heterozygous for the abnormal hemoglobin are able to cope with malarianell infection much better than individuals homozygous for the normal sickle-shaped RBCs. Hemoglobin, HbA. Thus, the natural selection tends to maintain the abnormal form of hemoglobin along with the normal form in a region where malaria is common. 7. Heavy metal resistance in plants. Plants have also produced some clear-cut instances of action of selection. Bradshaw, 1971, and Gregory, Antonovics and co-workers have studied varieties of several species of plants able to grow on soils contaminated with heavy metals, such as copper, zinc, lead. Such contaminated soils are often found near old mines where ores of these metals were exploited. The resistant varieties maintain themselves on soils with concentrations of the pollutants that are lethal or at least stressful for ordinary members of the same species. A conspicuous feature is that the resistant and non-resistant varieties may grow at distances only a few meters from each other, but meticulously confined to their respective contaminated and uncontaminated soils. Types of selection. Some individuals in a population are more successful than others in passing along their genes into the next generation. This may occur for many reasons. Some organisms die before they reach reproductive age. Some organisms fail to find a mate. Among those individuals that do breeding, there are differences in the number of offsprings produced. The result of these differences means that for one reason or the other there is a differential rate of reproduction among members of a population. We call this selection. Both in natural selection and artificial selection, discussed in Chapter 2, the principle is the same. Some organisms breed more prolifically than others, thus, increasing the frequency of some genes and decreasing the frequency of others. Both types of selection operate on the phenotype, not directly on the genotype. The following three types of natural selection have been categorized by population geneticists: directional, stabilizing, and disruptive diversifying. 1. Directional selection. The directional selection describes the change that occurs when a population shows a particular trend through time. In the foregoing discussion, i.e., examples 1 to 7 of natural selection, we have seen that the selection process favors individuals that are best adapted to new situations or to new ecological opportunities. Such selection is said to be directional since the norm for the population is shifted with time in one direction, figure 6.4b. The selection for low virulence that occurred over time among the myxoma viruses in the host rabbits exemplifies directional selection. The raw materials for selection were a series of gene mutations, each of which acted to reduce the virus's virulence. The curve of distribution shifted to the right, as the less harmful strains of viruses displaced the virulent strains. In fact, directional selection transforms the gene pool of a species or population toward the highest level of adaptedness that can be reached in the new environment. Apart from environmental change, origin of new and favorable mutant genes or supergenes brings into action directional selection. 2. Stabilizing selection. It describes the change when extreme individuals are eliminated from the population. So the intermediate values for a given trait are favored over the extreme values. The result of this process is reduced variability in the population. Most selection that occurs in populations is stabilizing and homeostatic, because it tends to maintain the status quo. As seen in figure 6.4a, the shape of the curve tends to narrow through continual elimination of the less adapted individuals. The birth weight of AM. AL stabilizing directional disruptive selection. AN. TNAA. AN figure 6.4. Schematic representation of three types of selection and their effects. Each curve represents the normal distribution of the character in a population. From top to bottom, the lower curves show the expected distribution after the impact of selection. A. Stabilizing selection. The intermediate values for a given trait are favored and preserved, and the shape of the curve narrows through the elimination of the carriers of the extreme value. B. Directional selection. The adaptive norm changes as less adapted genotypes are replaced by better adapted genotypes. C. Disruptive or diversifying selection. Two adaptive norms are generated when the population exists in a heterogeneous environment. After Volpe, 1985. Newborns provides a good example of a human character that has been subjected to stabilizing selection. Figure 6.5. The optimum birth weight is 7.3 pounds. Newborn infants less than 5.5 pounds and greater than 10 pounds have the highest probability of mortality. Given the strong stabilizing influence of weeding out the extremes, the optimum birth weight is associated with the lowest mortality. The curve for mortality is virtually the complement of the curve for survival. 3. Disruptive is diversifying selection. This form of selection occurs when the extreme values have the highest fitness and the intermediate values are relatively disadvantageous in terms of reproductive effectiveness. It is, essentially, selection for diversification with respect to a character. For example, shell patterns of limpets, marine mollusks, form a continuum ranging from pure white or dark tan. Limpets typically dwell in one of two distinct habitats, attaching either to white gooseneck barnacles, or to tan-colored rocks. As might be expected, the light-colored limpets seek the protection of the white barnacles, whereas tan limpets live by choice almost exclusively on the dark rocks. Limpets of intermediate shell patterns are conspicuous and are intensely selected against by predatory shore birds. If this disruptive type of selection favoring the extremes were to be accompanied by the sexual isolation of the two types of limpets, then two new species could arise. Evolutionary biologists also recognize the following two types of selection. Mean birth weight, optimum birth weight. 7.1. 20 equals U. L. F. A. 0.515 greater than OLLLL. Equals less than I. Section 5. EO. I. B. Underscore. A birth weight IB's B birth weight, LBS, figure 6.5. The distribution of birth weights of human newborns and the mortality of the various birth weight classes. The histogram shows the proportions of the population falling into the various birth weight classes. The mean birth weight is 7.1 pounds. B. The curve of mortality in relation to birth weight reveals that the lowest mortality is associated with the optimum birth weight, 7.3 pounds, after Volpe, 1985. A. Sexual selection. In many species of animals, females and males are strikingly dimorphic. Males are often larger, stronger, more brightly colored, have specialized song patterns and possessing various adornments, some of which may seem to be handicaps rather than advantages to their possessors. Females and males always differ in behavior to some extent, and sometimes strikingly so. Could sexual dimorphism originate in evolution by natural selection? Darwin found this improbable as a general explanation, and proposed in 1871 that sexual selection depends not on struggle for existence in relation to other organic beings or to external conditions, but on a struggle between the individuals of one sex. Thus, sexual selection is based on reproduction rather than survival. During the century since it was proposed by Darwin, the theory of sexual selection has met with even more opposition than the theory of natural selection. At present, sexual selection is that form of natural selection directly related to differential reproduction, including the finding and acquisition of a mate, copulation, fertilization and parental care. Huxley, 1938, had recognized the following two forms of sexual selection. 1. Epigamic selection. It is based on choices made between males and females. The selection by a female of a particular male as a mating partner as a function of his large rack of antlers or bright coloration would represent epigamic selection. 2. Intrasexual selection. It is selection based on interactions between animals of the same sex, usually between males. An example of intrasexual selection would be competition among males for a territory in a breeding ground. In practice, these two forms of selection overlap and distinctions are not clear, e.g., a given territory may contain better resources such as food, survival selection, as well as being more attractive to females, sexual selection, see Dewsbury, 1978. B. Group and kin selection. In a population under selection, the frequencies of some genes increase and those of others decrease as generation follows generation. The reproductive efficiency of individuals who carry the successful genes higher than that of the carriers of unsuccessful ones. What survives or dies are, however, not genes themselves but living individuals, who are their carriers. The question can nevertheless be asked whether, in addition to natural selection on the individual level, there can be group selection, i.e., selection of groups of individuals, such as Mendelian populations. Related species compete for resources that both are in need of, and one species may outbreed and crowd out another. For example, species introduced from foreign countries sometimes become pests or weeds and eliminate or greatly reduce the abundance of native species. This is particularly noticeable on oceanic islands. Island endemic species often seem to have reduced competitive abilities, and they are given short shrift. Group selection promotes the altruistic behavior of an individual which benefits its close relatives. This form of natural selection is known as kin selection. Altruism can be defined as a behavior that benefits other individuals at the expense of the altruist. If altruistic behavior is genetically conditioned, the Darwinian fitness of the altruists will be diminished, that of individuals who refuse to act altruistically will, on the contrary, be augmented. It would seem that natural selection would discriminate against altruism and favor self-preservation and egotism. Yet a population, colony, or tribe that includes many altruists may prosper more than a population burdened with egotists. Many kinds of behavior are equivalent to altruism, whereas they are really automatic and stereotyped. The behavior of honeybees, ants, termites, and other social insects has provided good example of altruism. Wilson, 1971. Individual, workers, toil selflessly and are ready to sacrifice themselves for the benefit of the colony to which they belong. The evolutionary development of this apparent altruism is easily understood. The workers and soldiers do not reproduce. They are sterile females or males with underdeveloped sex organs. The perpetuation of an insect society is the task of a sexual caste, the so-called queens and fertile males, which are a minority, sometimes single individuals, in the colony. Sexual individuals do little or no work after the colony is founded and display no particular readiness for self-sacrifice. The workers share genes with the sexual members produced in the colony. The result of this arrangement is that natural selection operates not in individuals but in colonies consisting of reproductive and sterile castes as units. The apparent altruism of the workers enhances the probability of transmission to the next generation of the genes these workers share with the sexual caste of their colony. RUAYRIO number 10. These ladybird beetles from the Chiricahua Mountains in Arizona show considerable phenotypic variation. Population. Genetics and evolution. 11. The individuals of a species constitute a population. The genetical studies for the inheritance of phenotypic traits in a given population is called population genetics. The population genetics is a quantitative science. To calculate the results of the mode of inheritance of genes in a given population, various statistical and mathematical models are employed in it. Certain fundamental aspects of population genetics are the following. MENDEIIAN population. A population of a particular species includes many inbreeding groups. The inbreeding groups may form a community within defined geographical boundaries and are called Mendelian population. A Mendelian population, thus, is a group of sexually reproducing organisms with a relatively close degree of genetic relationship, such as species, subspecies, breed, variety, strain, etc., residing within defined geographical boundaries where interbreeding occurs. Gene pool and gene frequency. To get if1 phenotypic ratio of a monohybrid cross, we began with two homozygous parental strains, such as AA and A. That is, we introduced the alleles A and A in equal frequency. A frequency is the ratio of the actual number of individuals falling in a single class to the total number of individuals. See Gardner, 1972. But in a Mendelian population, frequencies of alleles may vary considerably. For example, in Mendelian population of man, the gene for polydactyly is dominant, yet the polydactylous phenotype is fairly infrequent among infants. This indicates that frequency of the dominant allele in population is lower than that of its recessive allele and that both alleles do not exist in population in the 1:1 ratio like the individuals of monohybrid cross. Further, if all the gametes produced by a Mendelian population are considered as a hypothetical mixture of genetic units from which the next generatio YNLL arise, we have the concept of agen orgamete pool. The percentages of gametes in the gene pool for a pair of alleles A and A depend upon the genotypic frequencies of the parental MGen or trademark generation whose gametes form the T-Gagwifete pool O pool. Thus, if a population is of domi males MMMMSOPTEOITNCY pup, females _S nant genotype A, then the frequency equals NQ equals frequency of dominant alleles in the gene pool will Ma Yele bear relatively high and the percentage of gametes bearing the recessive A allele will be correspondingly low. Figure 7.1. The concept of gene pool after Stansfield, 1969. Two models of gene pool structure. Regarding the genetic structure of the population, the following two hypotheses have been proposed. 1. Classical hypothesis. It was developed by T. H. Morgan, 1932, and supported by H. J. Muller and Kaplan, 1966. The classical hypothesis proposes that the gene pool of a population consists at each gene locus of a wild-type allele with a frequency approaching 1. Mutant alleles in very low frequencies may also exist at each locus. A typical individual would be homozygous for the wild-type allele at most gene loci. At a very small proportion of its loci, the individual would be heterozygous for a wild and a mutant allele. Except in the progenies of consanguineous matings, individuals homozygous for a mutant allele would be extremely rare. The normal, ideal genotype would be an individual homozygous for the wild-type allele. According to classical hypothesis, mutant alleles are continuously introduced in the population by mutation pressure, but are generally deleterious and, thus, are more or less gradually removed from the population by natural selection. Periodically a beneficial mutant allele might arise, conferring higher fitness upon its carriers than the pre-existing wild-type allele. This beneficial allele would gradually increase in frequency by natural selection to become the new wild-type allele, while the former wild-type allele would be eliminated. Evolution, thus, consists of the replacement at an occasional locus of the pre-existing wild-type allele by a new wild-type allele, see Ayala, 1976. 2. Balance hypothesis. This hypothesis was proposed by Dobzhansky, 1970, and E. B. Ford, 1971. This hypothesis was derived by direct study of natural populations. According to the balance model, there is generally no single wild type or normal allele. Rather, the gene pool of a population is envisioned as consisting at most loci of an array of alleles in moderate frequencies. A typical individual is heterozygous at a large proportion of its gene loci. There is no normal or adel genotype, only an adaptive norm consisting of an array of genotypes that yield a satisfactory fitness in most environments encountered by the population. The proponents of the balance hypothesis argue that the ubiquitous allelic polymorphisms are maintained in populations by various forms of balancing natural selection. The fitness conferred on its carriers by an allele depends on what other alleles exist in the genotype at that and other gene loci. It also depends, of course, on the environment. Gene pools are codapted systems. The sets of alleles favored at one locus depend on the sets of alleles that exist at other loci. Evolution occurs by gradual change in the frequencies and kinds of alleles at many genealoci. As the configuration of the set of alleles changes at one locus, it also changes at many other loci. The balance model of genetic structure of populations has now become definitely established, although some controversy remains regarding the process maintaining the ubiquitous polymorphisms. Ayala, 1974, Lewontin, 1974. Chance mating or panmixis. Just as the gene frequency is controlled in the genetic laboratory, says the mating pattern, generation after generation, but outside the laboratory, mating is a chance or random affair. Every member of Mendelian population thus depends on chance or random matings. In other words, every male gamete in the gene pool has an equal opportunity of uniting with every female gamete. Zygotic frequencies expected in the next generation from such random gametic unions may be predicted from a knowledge of the gene allelic frequencies in the gene pool of the parental population. For example, the expected zygotic frequencies for allele A and of a gene pool can be determined by chance mating or panmixis. If P stands for percentage of alleles in the gene pool and Q stands for the percentage of alleles, the checkerboard of both alleles may predict possible chance combinations of A and A gametes as follows. Every member of Mendelian population depends on chance or random mating. Male gametes, P4. A, at. Female garetes, page. PQP, A. At P ill. Or P plus Q greater than equals P plus 2 PQ plus Q. Thus, page is the fraction of the next generation expected to be homozygous AA, 2PQIS the fraction expected to be heterozygous and G is the fraction expected to be recessive The zygotic combinations predicted in a randomly mating population may be represented by P, 2PQ, G%, where P represents the A genotype, 2PQ the A, and G the A genotype, or, in the form of equation, P greater than plus 2PG plus Q equals 1. When only two alleles are involved, and, therefore, P and Q represent the frequencies of all of the alleles concerned P plus Q equals 1. Since P plus G equals 1, P equals 1, Q. Now if 1, Q is substituted for P, all relations in the formula can be represented in terms of Q as follows. 1Q plus 2Q 1Q plus Q equals 1. Hardy-Weinberg Ya. The formula P plus Q greater than equals P plus 2PQ plus Q is expressing the genotypic expectations of progeny in terms of gametic or allelic frequencies of the parental gene pool and is originally formulated by a British mathematician Hardy and a German physician W. Weinberg, 1908 independently. Both forwarded the idea, called Hardy-Weinberg law equilibrium after their names, that both gene frequencies and genotype frequencies will remain constant from generation to generation in an infinitely large interbreeding population in which mating is at random and no selection, migration or mutation occur. Should a population initially be in disequilibrium, one generation of random mating is sufficient to bring it into genetic equilibrium and thereafter the population will remain in equilibrium unchanged in gametic and zygotic frequencies, as long as Hardy-Weinberg condition persists. Hardy-Weinberg law depends on the following kinds of genetic equilibriums for its full attainment. 1. The population is infinitely large and made at random. No selection is operative. The population is closed, i.e., no immigration or emigration occurs. No mutation is operative in alleles. N. Meiosis is normal so that chance is the only factor operative in gametogenesis. The significance of Hardy-Weinberg equilibrium was not immediately appreciated. A rebirth of biometrical genetics was later brought about with the classical papers of R. A. Fisher, beginning in 1918 and those of Sewell Wright, beginning in 1920. Under the leadership of these mathematicians, emphasis was placed on the population rather than on the individual or family group. Genetics was later brought about with N. Meiosis is normal so that chance is the only factor operative. No selection is operative. The population is closed, i.e., no immigration or emigration occurs. No mutation is operative in alleles. N. Meiosis is normal so that chance is the only factor operative in gametogenesis. The significance of Hardy-Weinberg equilibrium was not immediately appreciated. A rebirth of biometrical genetics was later brought about with the classical papers of R. A. Fisher, beginning in 1918 and those of Sewell Wright, beginning in 1920. Under the leadership of these mathematicians, emphasis A rebirth of biometrical genetics was later brought about with the classical papers of R. A. Fisher, beginning in 1918, and those of Sewall Wright, beginning in 1920. Under the leadership of these mathematicians, emphasis was placed on the population rather than on the individual or family group, which had previously occupied the attention of most Mendelian geneticists. In about 1935, T. Dobzhansky and others started to interpret and to popularize the mathematical approach for studies of genetics and evolution. Genetic equilibrium. As shown by Hardy and Weinberg, alleles segregating in a population tend to establish an equilibrium with reference to each other. Thus, if two alleles should occur in equal proportion in a large, isolated breeding population and neither had a selective or mutational advantage over the other, they would be expected to remain in equal proportion generation after generation. This would be a special case because alleles in natural populations seldom if ever occur in equal frequency. They may, however, be expected to maintain their relative frequency, whatever it is, subject only to such factors as chance, natural selection, differential mutation rates or mutation pressure, meiotic drive and migration pressure, all of which alter the level of the allele frequencies. A genetic equilibrium is maintained through random mating.